Hello everyone, I'm Simge Uçkun. Welcome to my graduation project presentation. My project name is PC12 Cell Lines Detection with Image Processing Approach, and my academic supervisor is Associated Professor Volkan Kılıç. I would like to show you the outline of my project. Firstly, I will talk about my motivation, then I will talk about the project states. Later, I will explain the image processing methods for determining the PC12 cell counts, branch counts, and length of all branches in a single cell. After that, I will present the final results. Then I will introduce you to our desktop application called PC12 app. And finally, we will review our conclusion and references. Let's start from the motivation part. PC12 has an embryonic origin from the neural crest that has a mixture of the neuroblastic cells and eosinophilic cells. PC12 cell shapes are round to ovoid and have numerous microvilli and small blebs. In this project, our motivation is developed an algorithm that can automatically detect PC12 cell counts, branch counts, and length of all branches connected to a single cell. I will talk about the project states now. In the first stage of the project, we created the cell detection algorithm by trying different image processing methods. Then we assigned labels to cells for future use in artificial intelligence approach. After that, we created the branch detection algorithm. And finally, we rearranged the pictures as a single cell and obtained the length of all branches with the pixel-based algorithms. Let's continue with the methods. In the cell detection algorithm, I followed two different paths over MATLAB and Python. Firstly, I will explain the MATLAB methods. This method introduced an innovative solution to detect cells by edge detection and morphology techniques within the powerful MATLAB image processing toolbox. Through the integration of edge detection algorithms such as Kenny or Sobel filters and combination with morphological operations like erosion and dilation, the proposed approach differentiates PC12 cells from the background noise. The Kenny edge detection algorithm uses a multi-stage algorithm to detect a wide range of edges, and the Sobel operator estimates the gradient magnitude and direction at each pixels in the grayscale image as shown in the figures. The Kenny edge detection computes of 5 parts, which are noise reduction, gradient calculation, non-maximum suppression, double threshold, and edge tracking by hysteresis. And the Sobel edge detection computes of 4 parts, which are converting the image into grayscale, convolving the gray image with Sobel X filter, convolving the gray image with Sobel Y filter, and calculating the gradient magnitude and direction. Image labeling is a type of data labeling that focuses on identifying and tagging specific details in an image. We created the cells without branch algorithm for labeling in MATLAB and labeled each of cells for future use. Morphological operations use a small binary image called structuring element. Here, there are several examples of structuring element. The structuring element is moved across the image and the pixels are compared with a reference value. Here are the erosion operation and dilation operation. The erosion operation removes the boundary of the objects in a binary image to make them smaller. It's used to separate objects by eliminating the noise and smoothing the boundaries. And the dilation operation adds a new pixel to the objects to expand their region. It can help to fill the gaps between the objects. In the cell detection algorithm, we converted the original PC12 image into binary by adding threshold. We scanned the background with the Strel disk function and extracted it from the original image. Then we created the dilated gradient mask by adding dilation to the picture. After that, we obtained a grayscale image with filters by using the infill function to fill the space inside the cells and the imclear border function to highlight the borders of the cells. Finally, using the connected component function, we counted each mask that not touch each other as a cell and reached the number of total cells. After that, I will talk about the Python methods. This method developed an algorithm for automatic detection of PC12 cells and branches. This Python script uses OpenCV, NumPy, Matplotlib libraries, scikit-image, and SciPy libraries. Contrast stretching method used in the Python script. This function modifies the intensity values of the increased contrast. A contrast stretching function is applied to enhance the image. This method used in the branch detection now. Skeletonization is a process that reduces foreground region in a binary image into a skeletal remnant as shown in the figure. Kernel or convolution matrix is a small matrix that's used for blurring, sharpening, edge detection, and other image processing functions. Essentially, this small kernel sits on top of the big image and slides from the left to right and up to down. Skeletonized image undergo convolution with a kernel to detect branch points in the cell morphology. After increasing the details of the pictures with the contrast stretching function, we divide the image into smaller, manageable subimages. Then we calculate the mean pixel intensity for each subimage, and if the mean is greater than a set threshold, the subimage is considered to contain a cell. This process is repeated for all subimages, and the total cell count is then obtained. Compute branch length function uses measurements.label to label the individual connected component in the binary skeleton image. In this context, a connected component can be considered as a branch of the cell. Each branch in the image is given a unique label integer and the total number of labels is stored. After labeling process completed, for each label, the function identifies its corresponding pixels using NumPy argvar function. For each pair of pixels in the branch, it calculates the Euclidean distance between them. This distance corresponds to the length between two pixels. The final length of the branch is sum of these lengths, as shown in the code. After the skeletonization process completed, the skeletons then undergo convolution with a kernel to detect branch points in the cell morphology. The convol function from SciPy is used for this purpose, and the total number of branch points is computed and stored. Our fourth part is results and discussion. Here we see the values of the number of cells and number of branches after applying our algorithms in different PC12 images. And here is the length of branch algorithm results for a single cell. Results show us the implementation of the image processing approach to calculate the number of cells, number of branches, and branch lengths showed promising results when applied to specific images under specific conditions. Our fifth part is custom designed desktop application called PC12 app. Firstly, when we enter an application, there is a login screen where specialists can log in with a special username and password. In addition, specialists can sign up for the application via the sign up screen. User information is stored in the SQL database program. After specialists log in the application, they can select the image file by clicking the file button in the upper left. PC12 image can be uploaded to the application through the file selection button on the main screen. Main screen divided to left and right to locate the image. The detection process can be started via the upper image buttons on the main screen after uploading the image. After the button is pressed, the process PC12 cells displayed on the right side of the main screen, besides the cell count and branch count shown in the upper side of the main screen. Let's watch a short video about the PC12 app. Here is the login screen. I entered my username and password. And here is the sign up screen. You can also upload the profile picture here. I'm selecting the pictures. Here is the displayed version of the pictures. Here is the binary image and the original image, and the number of cell counts and number of branch counts shown in the upper side of the screen. In the sixth part, we will review our conclusion. This project proposed to detect cell counts, branch counts, and length of branches on PC12 cell images using image processing approach that are compared for performance and integrated into a custom-designed desktop application called PC12 app. In conclusion, the implementation of the image processing algorithm for cell analysis in PC12 cells has demonstrated promising results when applied to specific images under specific conditions. The algorithm accurately identified individual cells and represent their morphological structure through the skeletonization process. Future endeavors will focus on enhancing the algorithm's adaptability to different image conditions through advanced noise reduction, contrast enhancement techniques, and dynamic threshold selection methods. By addressing these limitations, the algorithm has the potential to serve as a powerful tool for automated analysis of PC12 cell images, facilitating research in neuronal development and disease. And finally, you can see my references here. Thanks for watching.